Brain Atlas. Hello, I am Sergio.
Hello, my name is Alonso.
Good morning, I am Elisa.
Hello, I am Kim. I am going to introduce you to this topic. I am so excited. We will divide the topic in 3 different parts. First, definition and origin, then classification and uses and image analysis, and finally goals and examples.
But, first of all, what is Brain Atlas?
That's easy.
A brain atlas is a genetic geography of the brain composed of serial sections along different anatomical planes of the animal or human brain.
Moreover, brain atlases describe different aspects of brain such as anatomical, genetical or functional features and their relationships after applying appropriate registration and warping strategies.
That seems interesting and actual. But then,
How it all started.
in 2001 Paul Allen proposed the idea of three-dimensional atlas gene expression. The project was set in motion in 2003.
In 2004, the first atlas, the Mouse Brain Atlas, was released. Then, the final genome-wide dataset was released in 2006. And finally, the Allen Human Brain Atlas was made public in 2010.
Now, we can find different types of brain atlases.
Initially, Brain Atlas was intended to catalog morphological descriptions. Also, we can find atlases of brain structure based upon 3D tomographic images, anatomic specimens and a variety of histologic preparations. Moreover, there are atlases that include molecular contents such as myelination patterns, receptor binding sites, protein densities and mRNA distributions, and finally, some brain atlases are obtained from PET, functional MRI or electrophysiology. This last example describes the function.
However, single modality atlases may be insufficient because of the need to establish the relationship between different measurements of physiology and anatomy.
In response to these challenges, multimodality atlases combine detailed structural maps from multiple imaging sensors in the same 3D coordinate space. Multimodal atlases offer a realistic complex representation of brain morphology and function in its full spatial and multidimensional complexity. Now, we just need to know how we can obtain a multimodality brain atlas.
In order to obtain a multimodal brain atlas, digital mapping of structural and functional image data in a common 3D coordinate space is needed. Linear transformation, which includes rotation, scaling and translation, has some limitations. Local warping transformation, which includes local dilatations, contractions and shearing, would be a better option because it can adapt the shape of a digital atlas.
This deformation registration is achieved by a numerical method using an interpolator and an optimizer. In each step, the moving image, which is the image obtained from the patient, is transformed. This process is repeated until the optimum value for similarity between both images is obtained.
After this, we have our 2D registered moving image that matches with the general data and different regions of the brains are labeled. Then 3D reconstruction is produced by different algorithms and methods over the brain labeled maps. B-spline method is used as well as Marching Cubes algorithms. What are its goals?
Goals are simple. First, understand the connections between genes and brain functioning, to detect brain disorders. And second, research about neurobiological conditions and degenerative diseases.
Now we are going to talk about some mental health disorders and brain injuries.
Some examples are Pick's disease, motor neuron disease, Huntington disease, schizophrenia, Parkinson's, Alzheimer's and multiple sclerosis.
In this first example, images reveal severe focal shrinkage of temporal and frontal lobes bilaterally. The disease consists of progressive alteration in social behavior, which included apathy and occasional disinhibition.
Motor neuron disease develops muscle wasting and weakness. MRI often displays characteristic abnormalities within the corticospinal tracts in patients with motor neuron disease.
Huntington disease is a fatal genetic disorder that causes the progressive breakdown of nerve cells in the brain. It affects the basal ganglia of the brain.
Schizophrenia is a chronic and severe mental disorder that affects how a person thinks, feels, and behaves. The most common structural abnormality involves the lateral brain ventricles. These fluid-filled sacs surround the brain and appear enlarged. Parkinson's disease is a chronic disorder that affects movement. MRI data is used to look at the connectivity, or strength of brain networks, in the basal ganglia.
With Alzheimer, the brain displays many of the commonest features, such as brain shrinkage, or atrophy, and loss of function. Also, images reveal severe focal shrinkage of lateral occipital regions, associated with reduced perfusion which extended into lateral parietal and temporal regions. Multiple sclerosis provokes numbness and tingling in both legs and stumbling gait. MR images show high signal in periatrial white matter on T2 weighted images, and low signal on T1 pre and post gadolinium.
As conclusion, Brain Atlas is a really important tool for visualizing the brain in 3D, as well as its functions.
It allows us to study the brain functions, anatomical features and its behavior.
Therefore Brain Atlas makes doctors provide a better diagnosis and gives us a better understanding of the reaction of the brain to different diseases. That way, we get a better understanding of them.
It is only full of advantages,